It's great to be here today, and also my absolute pleasure to be leading this program, which is a large collaboration with many research partners here in New Zealand and also internationally. So today I'm going to be talking to you about how in the Healthy Trees, Healthy Future program, we're looking to understand how Phytophthora species impact their host plants. We've seen these images enough, and we all have different connections with the forest, but the one thing that unites us today is that we're all pretty passionate about avoiding photos like this in the future. But I do want to start today by acknowledging the importance of Kari to Maori, and the Healthy Trees, Healthy Future program is the first one under the umbrella of funding from the Kari Dieback program, which on the scientific side we're looking to actually have formal Maori engagement up front and to improve that relationship and to work in parallel with Maori moving forward. So we do acknowledge the importance of Kari to Maori and New Zealand. We also acknowledge that science offers tools to complement the established forest management and knowledge, and so that's what we're really presenting here today, is a suite of tools to better understand these symptoms and to put them alongside existing management. We are following a formal engagement process, and for this we're benefiting hugely from a lot of mentoring and guidance from people with far more experience in these areas than myself, and to which I'd like to acknowledge Phil's role he sits a bit in both the science and the Maori perspective, and I've learnt a huge amount, but also to the leadership of the Tangata Whenua Rōpū in being open to this process, and also in the learnings that we're having along the way as we partake in this process. So we're following the Te Araturaki process, of which Phil was a co-author, and it's specifically targeted at trying to teach us scientists to stop and have a bit of think about the Maori values that might be involved in our research. We're currently in dialogue with the Tangata Whenua Rōpū leadership, and we're looking to engage in a meaningful engagement with mana whenua. But I'm a pathologist, so I'm going to now step back into where I'm most comfortable. I've always worked on Phytophthora species, and I'm very passionate about finding real management solutions for them. But the scary thing is that worldwide, we know that there's 116 Phytophthora species out there causing significant plant disease. They are microscopic, we can't see them, and they're fungal-like, and they kill plants. And they're very aptly called Phytophthora, and that means plant destroyer in Greek, and that's pretty much exactly what we see them doing. Ian gave a fabulous introduction to this earlier, in which he noted the international examples of Phytophthora infestans, Cinnamomai and Ramorum, so I won't go into those further. But here in New Zealand, we have several Phytophthora diseases of significance in our plant systems. The one that I work most closely with is Phytophthora pluvialis, which causes red needle cast in radiata pine. This is interesting in that it's not affecting the roots of the trees that we know of, but it is affecting the needles. And obviously for wood production, we need needles on the trees because otherwise we don't grow wood. But Phytophthora are very significant in horticultural sectors. So, in apples, Phytophthora cactorum is a particular issue. And here's just a summary of a selection of a few of the Phytophthora species that are out there. So Phytophthora taxon agathis, Cinnamomai multivora are all in our native ecosystems, cactorum on apple, but then also down the bottom, pluvialis causing red needle cast, as I said, and Canovii are both very significant pathogens as well. Most of them are impacting the roots and collars of trees, but a few of them, as Peter quite aptly said a couple of days ago, have learnt to fly and are up in the canopy. So in the Healthy Trees, Healthy Future program, we want to really understand what all these phytophthora are doing to their different tree hosts. And so what we've set up is a project where we're looking at 3 different host trees, radiata, kauri and apple, and we're looking at what different phytophthora do to each of those. So we're using a multi-host pathogen model with those 3 species, and they'll be challenged by a total of 8 different Phytophthora species. And through this, we want to identify trees with broad resistance to a wide range of Phytophthora species, not just PTA, but the other ones that are out there as well. We want to develop screening assays, as Peter's just demonstrated, for identifying resistance in each of these hosts. We want to develop diagnostic tools, which Rebecca McDougall will talk a bit more about later on. And ultimately, we want to improve management of Phytophthora species. But along the way, we pathologists are also going to develop a greater understanding for what each of these pathogens is doing on the host and what the mechanisms of resistance are when we find them. So because we're looking at 3 tree hosts, we're able to take some of the learnings that we're starting to get from our productive Pinus radiata and apple plantations, and then start applying some of that knowledge through to kauri. So the project has led off with Pinus radiata, partly because we're based at Scion, but also we've got a really good breeding structure that Heidi Dungy heads up that gives us a system of breeding whereby we can track through the resistance far more efficiently. So over here on the left-hand side, we've got just a plate, and there's 48 different clones there that have all been screened. And what I've highlighted is that we've identified our 4 most susceptible clones in red and our 4 most resistant clones in yellow. And this gives us a really good group of clones that we can start asking those questions of what the pathogens are actually doing to them. When we take the resistant clones and screen them with Phytophthora pluvialis, you can see that the isolations of the Phytophthora over here on the right from a resistant clone is much, much lower than in our susceptible clones. At the same time, down in Hawke's Bay, Ian Horner is starting to look at apples in the same way. And you know, apples also have a very structured breeding population, so it gives us a great head start for asking some of these questions. So Ian's starting to screen the apple rootstocks that over the last 30 years they've been screening for Phytophthora resistance and taking the resistant clones and putting them into the rootstock breeding program without really actually asking the questions of what are the mechanisms behind that resistance. So now as part of the Healthy Trees, Healthy Future program, Ian and his colleagues are starting to ask those questions of why are these trees resistant? Are they going to be resistant to a range of Phytophthora species? Now, with both radiata and apple, the next step is to start applying some of the more modern Western science techniques to looking at the genetic and biochemical mechanisms of resistance. And what we're hoping is that if this is demonstrated on both radiata and apple, then it might be worth considering here in the kauri space. But our overall project model is to, as I said, take our 8 Phytophthora species and challenge them to each host. We're going to do that in both the roots and the foliage of the trees, look at the detailed pathology of the disease. So up here, I'm sorry, this is a bit blurry by the time we get here, but we've got the Phytophthora actually being produced, like pushed back out of the plant and continuing that disease cycle. When we start asking the questions of what genes are actually responsible for resistance, we need to know at what stage the Phytophthora is at in its attack of the plant. And by marrying up both the detailed pathology along with all of the genetic responses and the biochemical responses, we can start to get a much better picture of what's going on. So what does this all mean for the search for resistance in kauri? Well, Peter and Phil have given me a very good lead in here. But some of the fundamental differences with Kauri is that we're starting with a natural population. So we don't have those breeding lines and structured breeding populations that we do in apple and radiata pine. So we need to identify, firstly, if there is resistance to Phytophthora dieback. And ECHO studies are starting to show that we've got a difference. We just don't know what that means yet because it's still very much in an assay system. Then we need to start understanding the mechanisms of that resistance, and to do that, the toolbox of the genetic and biochemical markers will hopefully have been developed from radiata and apple and be able to start informing the Kauri work. As Phil said, it's then most likely that we'll need to screen widely. So we're looking to develop tools and markers that can be applied to that broad screening. And then if we do find resistance, it starts to present options for developing a resistance breeding program and increasing the level of that resistance in the standing forest. So as I said earlier, this is a wide collaboration with New Zealand partners. So it's being led by us at Scion, but clearly involves research from Landcare, Plant and Food, the Kauri dieback program, and all of the partners that you see there. So just acknowledging them. If you'd like any more information on this and to keep tabs of the program, we have another 5 years of funding and a website that you're very welcome to go and visit, but also you can sign up there for a newsletter. So if you'd like to keep informed on a quarterly basis of where we're at in this research, then I very much welcome you to. Thank you very much.